Hello, good afternoon, everybody. Welcome to our webinar, The Introduction to Single-Cell Sequencing. My name is Katarina Pankova. I work at Singleron as the Associate Director of Marketing. And I'm very happy to be here today and have this opportunity to introduce you to single-cell sequencing and the technology that we have developed. So let me start with the presentation that I have for you. And first of all, I would like to give you a brief overview of who Singleron is as a company and what we are trying to achieve. So Singleron Biotechnologies is present globally at different places around the globe. So we have our European headquarters in Cologne, Germany. We also have a small office and the lab in the Connecticut in US. We are present in Singapore and in China as well. Now, we are a company that is dedicated to single-cell sequencing, and this is our specialty. And the question, of course, is why single-cell sequencing? Why is it so important, and what it actually can give you and your research, how it can advance your research? So there is a major difference between the bulk sequencing and the single-cell sequencing. When you do bulk sequencing, bulk RNA sequencing, you usually start with your tissue as an input material. So you usually have mixed types of cells in the tissue that you are trying to investigate, and you extract your RNA from this tissue, and basically you pool all the RNAs from different types of cells together. So once you sequence a library that was basically created from this RNA pooled from various types of cells, you get an average profile of the entire sample. So you are not able to distinguish which kind of genes were expressed to higher levels in different types of cells, because what you get is the average of everything that you had in your sample. And this is a major limitation that bulk RNA sequencing actually has. And exactly for this reason, a single-cell sequencing has been developed. So the major difference between the bulk sequencing and the single-cell sequencing is that as an input in single-cell sequencing, you take the actual individual cells. So basically, you don't start with the piece of tissue, but you dissociate this tissue into individual cell types, and you extract the RNA from each of these cells separately in a very, I would say, elegant manner that I will explain in a detail a little bit later. So basically, you get your RNA from each of these cells separately, you label this RNA, and then you create one pooled NGS library which you then sequence, and you can get the information about the gene expression in the individual cell types separately. So basically, you can assign the gene expression to the individual cell types which were present in your sample. And this is of great importance because the majority of tissues that you most likely work with do not comprise only one cell type. It's almost always a mixture of different types of cells. And in order for you to understand which genes are expressed to what kind of levels in these different types of cell types, the single-cell sequencing, I think, is the key. So single-cell sequencing can be applied basically to any type of tissues that show variability or heterogeneity in them. And the fields that benefit most from single-cell sequencing are the cancer research, neuroscience, but also immunology, developmental biology, and basically any other research field that deals with tissues comprising of different types of cells. Now, single-cell sequencing has been around for quite some time. It has been named the breakthrough of the year in 2018, and it also has been named the method of the year in 2020 by the journal Nature Methods. The number of publications that have used single-cell sequencing has grown rapidly. So in the past year, there's been more than 3,400 research papers that utilize single-cell sequencing, and we believe that this number will be growing in the further years even further. So it might well be that in a couple of years, the single-cell sequencing will become a ubiquitously present method that is basically that can be utilized in any lab capable of producing NGS libraries. The single cell sequencing, in Singleron, we are experts in single cell sequencing. And we have a lot of experience that comes from working on different research projects. So far globally, we have worked on more than 2,300 research projects. We have served more than 600 different organizations. And as an interesting fact, we have processed more than 300 million single cells. And our expertise comes from working on different types of tissues, as preparing the single cell suspension is the first most crucial step in your single cell sequencing experiment. So we have experienced processing of more than 660 different sample types, which come mostly from a human, mouse, and non-human primate tissues. And our success rate is at 94%, meaning that we can successfully process vast majority of tissue samples that arrive to our service labs. The single cell sequencing, when we look into the history, how it has developed, it originally started exactly as it sounds, so with single cells. In 2009, the single cell sequencing was established in a publication by Tank, and it was actually really the single cell that has been sequenced. And since 2009 until today, there has been a tremendous amount of work and that has been done, and tremendous development in the technologies that are available. So now when we are talking about single cell sequencing, we actually don't talk about one single cell being sequenced. In a single experiment, we can process thousands and thousands of individual cells. So currently with Singleron's technology, you can process up to 30,000 cells in a single experiment. So just imagine like how much data that is and how much information you can get just with a single experiment that you perform. So there has been a variety of different methods for single cell sequencing with varying throughput. So as I mentioned, in 2009, you had this throughput of one single cell, but currently Singleron is capable of achieving much more in a single experiment. And let me briefly introduce you to Singleron's technology and how single cell sequencing actually works. So the core of our technology is called a SCOPE chip. So SCOPE chip is this small device that you can see in this image. It's basically a microfluidic chip that has two openings. And it's a single-use device that has two openings for pipetting. And it's basically of a size of a microscopic slide. And you can easily pipette in and out individual reagents. You don't need any dedicated instrument to process this microfluidic chip. You can do your single cell experiments just with a P200 pipette that you already have in your lab. The chip itself is covered with microvels. So in the standard configuration, you have 150,000 microvels between the two openings of the chip. So basically the area between these two openings is covered with microvels. And basically how it works is that you prepare your single cell suspension and you load it onto the chip. And on this chip, individual cells fall into the individual microvels. Once this step is accomplished, you load the barcoding beads. So again, you use this opening on one side of the chip, and you load the barcoding beads, which are labeled here as black balls, black circles. And you basically get one barcoding bead falling into each of the microvel. So what you are aiming for is to get a single cell and the single barcoding bead to meet in one microvel. This is the situation that you want to achieve. And once you have the single cell and the barcoding bead that would meet, you introduce the lysis buffer. Basically, the cell ruptures, the cell membrane is ruptured, and you capture your mRNA from the cell onto the barcoding bead. Now, once this step happens, you can collect the barcoding beads from the microchip and create the sequencing-ready NGS library in the following steps. Now, people often ask, how come that we ensure that in each microville you only have one single cell? And the key to that is that we underload the chip. So basically, we have 150,000 microvilles, and we only capture 10,000 cells in the standard configuration. So basically, it means that the majority of the microvilles actually remain empty, and the ones that have cells in them would very likely only have a single cell in each microville. Now, the question also is, how do we ensure that we have only one barcoding bead in every microville? And the answer to that is, it is the size of the beads. So basically, there is not enough space for more than one bead to occupy each microville. So basically, you can only get one bead per microville. And this is how we achieve the situation that the bead and the cell would actually meet in the same compartment. Now, what exactly happens when the bead and the cell come together? So these beads are covered with oligonucleotides. They have a lot of oligonucleotides on their surface. There are millions and millions of oligonucleotides attached to them. And every barcoding bead has the following sequence on them. So first of all, they have this green poly-T tail. This sequence is important for the capturing of the mRNAs. So as you know, the mRNAs have the poly-A tails. They are polyadenylated, which basically this poly-A tail prevents mRNA degradation. And this poly-A tail is basically what we capture using our barcoding beads onto the oligonucleotides which are on their surface. Another important sequence on the barcoding beads that we have is so-called cell barcode. And it's important to mention that this cell barcode differs between every single bead. So basically, this sequence is the key for identification of all the mRNAs that come from the same cell. So during the bioinformatic analysis, once we label the mRNAs with the cell barcode, afterwards we put them together, and during the bioinformatic analysis, we can again split them into the individual cells from which they actually come from. So we know that all the mRNAs which would have the same cell barcode came from the same cells. So this is basically the trick how we do that, how the single cell sequencing works. Another sequence that is important is so-called UMI. It stands for Unique Molecular Identifier. Basically, we use every UMI that you have on the barcoding bead has a different sequence. So basically, every oligo on the barcoding bead would have a different UMI sequence. And we use this UMI sequence to basically count how many mRNAs or how many transcripts, how many reads will be assigned to every gene that we actually count for. So this UMI sequence serves for a quantification of transcripts during the bioinformatic analysis. And lastly, what we have on our barcoding beads is so-called PCR primer. So basically, the sequence is important during the cDNA amplification, the library preparation, and during the sequencing. So how our single cell sequencing workflow actually looks like. First of all, you start with a piece of tissue, which you dissociate into single cell suspension. Now, when you want to handle this piece of tissue, sometimes it might happen, especially if you work with the clinical samples, that this piece of tissue has to be transported to a lab that actually enables you to do the single cell sequencing. And in such instances, you might need to preserve your tissue for a certain amount of time. And we have a solution for that. We have developed a buffer, tissue preservation buffer, which enables you to store your tissue and basically ship it wherever you need to have it shipped within 72 hours at 4 degrees. So you can easily just drop your piece of tissue into the vial that we provide and send it to where it needs to be processed. Then once the tissue arrives to its final destination, you can proceed with dissociation. And for that, we also have a dedicated solution. So we have so-called SLife tissue dissociation master mix. It's basically an enzymatic solution that enables you to efficiently create a single cell suspension from a variety of different tissue types. And we have data showing that the cell viability that you achieve when you use our tissue dissociation master mix is very high. To create the single cell suspension using our dissociation master mix, you don't need to have a dedicated instrument. You can put your tissue with the enzymatic mix into a thermal bath, and basically this is all you need. But in case you want to have it automatized, we also offer Singleron Python instrument, which is an automated tissue dissociator. It might be of interest to those of you who process a lot of tissues and do a lot of experiments and wish to have some of the workflow automatized. So we offer this option as well. We have a solution to dissociate your tissues in the automatic fashion. Okay, so we are at the step where the tissue has been processed and we already have our single cell suspension. And what we do next is that we simply load this single cell suspension onto the chip. And the chip itself you are able to visualize this chip on your own under a simple microscope. So basically you can see how the microvels, the empty microvels look like. Once you load the chip with your single cell suspension, you can see a proportion of microvels which are occupied with single cells. You can easily visualize it under the microscope. And lastly, when you load the microchip with the barcoding beads, you can visualize this as well. And you can see whether your loading of the chip was actually successful and whether you should proceed further with your experiments. Now, for the loading of the chip, as I mentioned, you do not require any specialized equipment. You can always do it simply with a pipette. That's not an issue. For those customers who wish to have this step automatized, we have a dedicated instrument called Singleron Matrix. And this basically saves you some pipetting steps and enables you to process 2 chips simultaneously in the automatic fashion. So once the processing of the chip is finalized, you can basically pull your beads with bound mRNA from the microchip, and you can proceed with the reverse transcription, cDNA amplification, and the library construction. Now, in this picture here, I wanted to show you how does loading of the chip actually look like in the lab. So on the left image, you can see the chip, and basically you can see how you use a pipette to load your single cell suspension onto the chip. Now, if you are new to our products, you can always ask to get you some training kit where you basically practice the loading of your single cell suspension and the barcoding beads before you do the actual experiments. So you can get yourself trained before you start with these experiments. What is important to know is that, for instance, the position of the pipette should be always at 90 degrees. You should pipette very slowly, and you should have a dedicated pipette tips that fit the opening of our chip. So, of course, we provide all this information in the user guides that come together with our products. For the customers who wish to have this processing of the microchip automatized, we offer a solution. So on the right portion of the slide, you can see an image how you basically load the microchip when you use our automated matrix processing system. So you simply load your reagents into the cartridge that we provide. You just close the instrument, press run, and it offers a walkaway level of automation. And it basically it processes the chips, up to 2 chips simultaneously in the automatic fashion. Okay, so when the chips are processed, the product that you get from these chips are barcoding beads with the mRNA attached to them. And now you need to do the reverse transcription to actually produce the cDNA. You need to amplify the cDNA, and you need to prepare an NGS library for the sequencing. Once you have that, our products enable you to create libraries compatible with the Illumina sequencers. And then you can proceed, once you have your sequencing data ready, you can proceed with the bioinformatic analysis. For this bioinformatic analysis, we offer 2 different solutions. So one is called CellScope software, and the other one is called Synecosys database. So now let me briefly show you what kind of data you will get from our single cell sequencing products. So when your data from the Illumina instrument arrive, you get raw data in the FASTQ format, and you can easily use our CellScope software. It's a pipeline that is free for you to download from the GitHub, and you just feed in your raw data into this pipeline. And as a result, you get the expression matrix. So basically, you get a file that tells you for what's the gene expression for every single gene, every single protein encoding gene. So this might be up to 20,000 different genes in humans for every single cell that was processed in your experiment. So as I already mentioned, in one experiment, you can process up to 30,000 cells. Meaning that this expression matrix will have a lot of data in there, because you will get this gene expression counts for every single gene, for every single cell that is that was on your chip. So basically a lot of data. And the way how we normally look at this data is to do so-called data clustering. So you might have already seen these types of plots before. Basically, what these plots show you is how your single cell sequencing data look at the first glance. So you should know that individual dots that you have on this plot represent individual cells. And if the 2 cells are close to each other, it means that they have similar kind of gene expression. So based on the position of these dots, which represent cells on this plot, you can get an idea what kind of, how many clusters, how many different types of cell types you actually have in your data set. So once, this is the output that you get using the CellScope software. And what we also offer is the secondary analysis using the Synecosys database. So Synecosys database enables you to do a very intuitive secondary analysis of your data. It enables you to perform automated annotation in order to identify individual cell types based on the previously reported marker genes from the scientific literature. So basically, you will, once you process your data with the Synecosys database, in the Synecosys database, you will know that, for instance, this red cluster of cells would correspond to B cells. This small green cluster of cells, I think this would be the macrophages, and so forth. So we would basically identify the cell types that correspond to the individual clusters in your data. In addition to that, the Synecosys database also offers you to perform secondary types of analysis of different sorts, such as, for instance, the trajectory analysis that you see here. Now, what we offer in Singleron are the kits that enable you to take tissue as an input and produce a sequencing-ready NGS library as an output. And then we provide you with the tools necessary to do the bioinformatic analysis yourself. As I already mentioned, you don't need a dedicated instrument to perform single-cell sequencing experiments. If you want, we offer you solutions to automatize the workflow. So you can get your single-cell tissue dissociator, Singleron Python, or you can get the instrument to automatize the chip processing. But this is not a necessity. So it's only up to you whether you wish to use these instruments or not. So what you actually need to have in your lab in order to start with the single-cell sequencing experiments. So in this workflow here, I'm showing you a little bit more detail what happens during as you prepare your materials, your single-cell sequencing NGS libraries. So as we already mentioned, you start with the tissue, you create single-cell suspension, you load the suspension onto our chip, and then you collect the beads with the mRNA, and you process with you proceed with the reverse transcription. You amplify the cDNA that you obtained, and then you proceed with the library preparation. Now you can see that this whole workflow is split into two parts. So basically, in the pre-PCR part and the post-PCR part. So why is that important? So if you haven't done any NGS libraries before, you should know that if you do PCR, there may be a lot of DNA, a lot of PCR products up in the air. And if this kind of PCR products can contaminate what you put into your PCR mix, which you are preparing. So basically, if you do not split the pre-PCR workstation from the post-PCR workstation, you could get some PCR product contamination from the previous runs. And this is why we recommend to have the pre-PCR workstation, the post-PCR workstation in the separated labs. And you should also have your dedicated lab code and basically all the instruments and consumables dedicated either to the pre-PCR or the post-PCR lab, and do not mix things between these two workstations. So in the pre-PCR lab, you should have, if you start with a tissue, you would need the electronic scale. You would need to basically weigh your tissue. Our products work with roughly 100 to 200 milligrams of tissue. So this is the input that you should use. And when you are preparing your single-cell suspension from the tissue, you would also need to have a refrigerated centrifuge in order to basically prepare high-quality single-cell suspension. Apart from these two instruments, as we already mentioned, you would need a thermal mixer. So this is where you incubate your tissue with the enzymatic master mix in order to prepare the single-cell suspension. And then you would just need the regular stuff that you already have in your lab. So the tabletop vortex, a small centrifuge, and the microscope. The microscope you would need in order to assess the quality of your single-cell suspension, and basically to determine the concentration of the single-cell suspension that you will then further load on the chip. So these are all the instruments that should be in your PCR lab. As I already mentioned, there is probably nothing that you already wouldn't have in your lab. So basically, it's the standard equipment that you most likely already have on your benchtop. Now in the post-PCR lab, so once you prepare your cDNA, you again should have the vortex and the tabletop centrifuge. You should have a PCR machine because you will perform the cDNA amplification. And then if you already have done any type of NGS libraries, you might have these instruments, or you might have access to those instruments in your organization. So you would need a fluorometer, for instance, such as qubit, in order to determine the small amounts of the DNA that you will basically produce in your library preparation steps. And you would need to have a tape station. So this is an automatic electrophoresis station that will enable you to do the quality control steps during the preparation of the NGS libraries. Okay, so this would be the instruments that you actually need to have in your lab. And of course, if you want and you have some spare space on your benchtop, we would be happy to provide you also with the tissue dissociator and the automated chip processing system. But this is not a necessity. So you can always do your single cell experiments just with what you already have in the lab. You can load the chip manually. That's not an issue. So how does the single cell RNA-seq workflow actually look like? So in this table here, you can see the estimated times that you would need for each step. So if you start with a tissue, it would take you, depending on the type of tissue, between half an hour to 1 hour to dissociate it into single cell suspension. In case you work with blood samples or PBMCs, you can basically skip this step because you already have your single cell suspension. And then you would need approximately 1 hour to basically operate this chip to load your single cell suspension, load the barcoding beads, and basically then collect it from the chip. And then you would need to have approximately 1.5 to 2 hours, depending on how fast you pipette to get your reverse transcription reaction done. So once this step is finished, you can have a break. You can safely store your cDNA in the fridge, and you can decide whether you want to store it, or basically you can also do this protocol as a one-day protocol, which can be finished between 7 to 9 hours. So majority of the work majority of the time that this workflow takes is basically the cDNA amplification and the library preparation steps. And there are several opportunities to stop this workflow once you have your cDNA. So there are multiple possible breaks. So you don't have to do this whole workflow within 1 day if it doesn't simply fit your schedule. It's quite flexible. Okay, so far I have talked to you about the products that we have. For those of you who wish to do their experiments themselves in the lab. At Singleron, we also offer possibility to outsource your experiments to our service lab. So basically everything that we have spoke about until now, so from tissue through single cell suspension, chip, and basically creation of the libraries, sequencing, and the bioinformatic analysis, we can do all these steps for you in our service lab. So how that works is that you simply send us your tissue and we take care of the rest. For the projects with over 50 samples, we also offer bioinformatic analysis, but yes, this only starts with projects of samples of more bioinformatic analysis is only included in the projects of with higher number of samples. So not every project would include the bioinformatic analysis from our part. So in case that you would like to outsource your experiments for whatever reason, you don't have to do single cell sequencing in your lab. You can just send us the samples and we will take care of the rest. How that works is quite simple. So as I already mentioned, we have this tissue preservation buffer. We simply send you the vials with this preservation buffer, and you drop your piece of tissue in the buffer and send it to us at 4 degrees. It has to reach us within 72 hours. This is the time when the tissue can be, for how long the tissue can be stored in the buffer. And we basically take care of the rest. So we assess we do the tissue single cell dissociation and basically all the processing, the whole single cell sequencing experiment for you. Okay, so I can see that you have some questions, and I would also like to introduce you to our Multi-Omics solutions, but I think it would be better if I took your questions now, and then I can continue in the presentation further. So let me check the questions that you have in the chat. So one question is that, is the chip any different to a flow cell? You probably mean by flow cell a chip that you actually put into your Illumina instrument in order to get your libraries sequenced. So in that situation, yes, our chip is something different. So our chip enables you to prepare NGS libraries, or is the first step before the preparation of the NGS libraries, but you would still need to sequence those libraries on the Illumina instruments. So yes, there is a difference between our chips and the flow cell. How do we make sure that only one bead binds to the primer? Okay, so basically the question is on what is actually on our beads. So every single bead is covered with millions and millions of different oligonucleotides which are on their surface. So basically we are trying to capture as much of the mRNA as we possibly can using the poly T tails on the barcoding beads. Another question, has the single cell RNA sequencing been used in the plant science so far? So yes, we have used single cell RNA sequencing in the plant science. So we already have data from the Arabidopsis rosettes, as well as from the Arabidopsis root. So this data come from the protoplasts. And what we see as an advantage is that the protoplasts they are very fragile. And as they fall into the individual microvelles, they fall gently by gravity. So we believe that the microvelle-based method for single cell sequencing is advantageous because you preserve you preserve these protoplasts. You do not damage them. RNA integrity and the output content. So I can share this information with you later on. I don't have the slides on the RNA specific on the plant specific data in my current presentation, but this is something that we can share with you. And another question is that whether we have a machine learning algorithms to annotate the cell types. So at the moment, what our Synecosis database uses is the cell ID, which works for the human, mouse, and non-human primates. So for the plants, I think that might be a little bit tricky. Another question is, in case of automation of mRNA, how do you make sure that you are dealing with a single cell without the chip? So basically, we collect the barcoding beads which have mRNA attached to them, and we remove them from the chip. So this is how it works. And then we just convert the mRNA into the cDNA. Another question is that long non-coding RNAs also have polyase. So how do you make sure it's the pure mRNA we are dealing with? It doesn't have to be pure mRNA. So everything that has poly-A gets attached to our barcoding beads. It's not an issue. So everything that is polyadenylated gets attached. The technology can be, the question is whether the technology can also be used for plant cells. The answer is yes. We have materials on our website showing you what we can do with the plant cells. And the last question we have is, how are the beads prepared? Can the primers be changed to cover different domains in a single run? Yes, and this is exactly what brings me to the next part of the presentation. So I would like to show you the multi-omics capabilities that we have and what different types of oligos can be attached to our barcoding beads to basically provide them with new capabilities. So our product can be used either to do the transcriptome profiling of single cells. We can also do the single nuclei, or we can profile yeast cells, which are specific because they need special kind of digestion of their cell wall. We also have possibilities to do the transcriptome profiling combined with the VDJ profiling, in case you are interested in immune repertoire profiling, repertoire profiling, or the full length information on the T cell and B cell receptors. We do transcriptome profiling of single cells combined with the target sequences. And we also can combine the transcriptome with temporal resolution or transcriptome with glycosylation on the single cell level. So let me start with the single cell versus single nucleus. Basically, single cell sequencing utilizes all the mRNAs that are in the cells. And depending on the type of tissue, you can detect on average between 1,000 to 5,000 genes per cell on the average. And it's very good in detecting the immune cells in case that you are working with T cells, B cells, B cells, or NK cells. This would be the preferred method of choice. And the fresh tissue, which you can use for such experiments, can be stored for up to 3 days in our tissue preservation buffer. On the other hand, some people tend to use single nucleus sequencing, and this is mostly for the tissues which have been frozen, because during the freezing process, you disrupt the cellular membrane. So in case that you have some samples in your freezer and you want to do single cell sequencing on them, single nucleus would be preferable. So of course you get a lower amount of material because you have less of mRNA into the nucleus, but it's possible. You basically get something between 500 to 2,000 different genes per cell. And also it works on the tissues that are difficult to dissociate, such as, for instance, the brain, muscle tissues, adipose tissue, or pancreatic tissue. And yeah, advantage is that frozen tissue can be stored long-term. So basically, you can still do your single cell experiments on the tissues that have been frozen in the past. Now, let me get to target sequencing. So why do people actually offer target sequencing, and what does it actually mean? So as I already explained on the beads, you have this sequence that contains poly-T sequences, unique molecular identifier, the cell barcode, and the PCR primer. And basically you use this sequence, you use the poly-T sequence to capture the mRNAs. So on the picture below, you can see actually the bead with this oligo attached to it. I have to say that there are millions of these oligos, so only one is shown in this picture. And in the black, you can see the mRNA that is being captured by the poly-T tail. And as you capture it, in the next step, you basically convert this mRNA into the cDNA, which has this cell barcode and everything that was attached to the bead now attached to the cDNA. So basically you convert your mRNA into the cDNA and you label it with a cell barcode. Now, you should know that the mRNAs that you usually have are quite long. So basically they can be on average, I think in human, between 1,500 kilobases up to 2,000 kilobases long. And such long mRNAs, you cannot easily sequence on the Illumina instruments because Illumina offers short read sequencers. So basically, once you have this cDNA, you need to fragment it in smaller pieces. And now what happens to this cDNA when you fragment it? You fragment it in random places. So basically you would get random fragments of this cDNA, but you cannot use all the fragments. For the sequencing, you can only use the fragments which have the cell barcode attached to them. So basically, you can only use the 3' end fragments because these are the fragments that have the cell barcode, which is crucial for you to understand from where the mRNA comes from. So basically, you only select fragments from the 3' end part. And this is why in this graph that shows the coverage along the gene, this is why you see this high peak at the 3' end of the gene body. So on this graph on the right side, on the x-axis, you can see the gene body on the x-axis. So basically, let's imagine that you have an average type of gene with its 3' end and the 5' end. And on the y-axis, you can see the coverage. So basically, how many reads, how many reads you basically get along this gene length when you use single-cell sequencing. And you can already see that you get like a very high number of very high coverage at the 3' end of the gene because of this 3' end bias. So basically, due to fragmentation, you end up in a situation where you select your fragments at the 3' end, and this is why you see this 3' end bias. Now, it wouldn't be a problem because you can still identify the genes from which the transcripts come from. You can still profile them and determine the gene expression. But the problem comes if you are interested, for instance, in a mutation that is further away from the 3' end. So if your mutation of interest lies closer to the 5' end of the gene, and you still want to have it sequenced because it's important for your research, it becomes tricky. And this is why we offer target sequencing with beads which have been specialized to the research question of your choice. So basically how this works is that we use the very same technology, very same microchip, but now the beads look different. So basically we not only have the poly details that capture the transcriptome, but we have probe specific to the genes of our interest. So basically these probes can capture genes by the middle of their sequence, bring them closer to the barcoding beads, and get the sequence of your interest sequence such that you detect your mutations, gene fusions, or whatever you are interested in, which lies further away from the 3' end. So we can, with this method, we already have pre-constructed panels for the Epstein-Barr virus detection, lung cancer, blood cancer, also for the clonal hematopoiesis of indeterminate potential. But you can always create your customized panel of up to 10 genes in case you are interested in sequences which lie further away from the 3' end. Also, another advantage is that you can, for instance, detect the viral sequences. So you can capture them and ensure that you enrich for them. Because it often happens that you might be interested in transcripts which are expressed to low level. And this is something that FocusScope can also help with. So it also enables you to enrich for the transcripts of your choice already on the level of the barcoding beads. This is an example of the advantage of the FocusScope that you get to when you are trying to, for instance, detect the viral sequences. So in the left portion of the image, you can see that if you only use the poly-T probes to capture Epstein-Barr viral sequences, you can see that the number of cells in which you detected the virus, depicted here in red, is rather low. Whereas if you use a dedicated FocusScope kit, which enables you to enrich for the viral transcripts at the very beginning, you can see that this number of cells positive for the viral sequences is much higher. So we can see that this detection rate goes up more than twofold. And this is the advantage of the FocusScope with dedicated probes to capture or enrich the genes of your choice. Okay, I think we can cover one last topic, which is the multi-omics and the glycosylation. So we have dedicated products that not only enables you to detect transcripts, but also the glycosylation on the cell surface. And how it works is that you add a specified ProMOSCOPE tag on the top of your cells. So basically you have a ProMOSCOPE tag that binds to the certain sugar residues onto the cell surface. And then these ProMOSCOPE tags behave as an mRNA because they have the poly-A tail. In the next step, when you basically load your tagged cells on the chip, you capture this tag from the cell surface onto the barcoding beads. So just imagine that you label the top of your cells with the tag of your choice that basically enables you to detect the glycosylation on the cell surface. Then this tag behaves as an mRNA, and together with all the other, together with all the other mRNAs present in your cells, you not only capture them, but also this tag which you basically added there artificially. And you can quantify how much of this ProMOSCOPE tag you had for every single cell. And in the end, you can get the information of how much glycosylation is there on the cell surface of every single cell that you analyze in your single cell sequencing experiment. So this is an example of the data that you can get. Basically, this comes from the bone marrow. So you can already see this UMAP plot where every single dot represents a single cell. We have different types of cell populations in this data set. And on the middle portion of the image, in the red dots, you can see, depending how red the color is, the more red, the more glycosylation was present in the cells. So you can, for instance, see that the monocytes had on their cell surface quite consistent glycosylation in the whole population of monocytes, whereas for other types of cells found in the bone marrow, this glycosylation pattern or glycosylation amounts were quite different. So this is an example of a type of multi-omic approach that you can also combine, or you can also utilize in your single cell sequencing experiments. Okay, so thank you very much for your attention. This is everything that I wanted to share with you today. I hope that I didn't overwhelm you. And I will try to get to a few more questions that I got from you. Yes, so the question is whether we perform differential analysis if all cell types are preserved equally in the preservation buffer. So for the preservation buffer, we have data showing that the cell viability is not really affected if you store your tissues in the buffer for up to 72 hours. And as for the neutrophils, neutrophils are tricky. So with our microbial-based method, you can most certainly sequence the neutrophils, but our preservation buffer doesn't work on the single cell suspensions. So basically, if you want to sequence the neutrophils, you would most likely not store them in the preservation buffer because that one is only dedicated to solid tissues. Another question is whether we can fragment and then make cDNA to avoid a 3' bias. So with this 3' bias, there is always this problem that you have to keep your cell barcode attached to your cDNA, to your fragment. So yeah, I mean, with the short read sequencer, it's quite difficult to avoid this 3' bias. And another question is whether we can expand on the liquid handling robot used in single cell analysis. So the advantage of our approach is that it can be automatized. We offer instruments that enable you to do the automation. And it depends what kind of liquid handling robot you would like to use, because you most certainly don't need any automation to separate the individual cell types. So these kind of instruments you don't need, because with our matrix instrument, you simply get the separation of cells in the microvells on the chip. You get mRNAs from the cells labeled, and then you simply collect them. Okay, let me see if I got all your questions, because I think I'm in the end, just to make sure that I didn't miss anything. No, so that's it. Thank you very much for joining this webinar today. I hope it was useful. If you have any questions, please feel free to get in touch with us, and I'm looking forward to meet you in the next webinar in the future that we might have. Thank you very much.